Hi there, Dr. Biology here. This is part 4 of my respiration videos. This one's going to look at anaerobic respiration. So just to recap just quickly, we've already seen that with aerobic respiration oxygen is needed if the hydrogen atoms produced in glycolysis and the Krebs cycle are to be used in the production of ATP. So now we need to look at what happens when oxygen is temporarily or permanently unavailable, and this is when anaerobic respiration will come into play. Now what's quite interesting is that anaerobic respiration, the processes are slightly different in terms of what the products are that are produced in plants and microorganisms compared to animals, so we'll look at those differences as well in this video. So in aerobic respiration, obviously oxygen is important, so the hydrogen atoms produced in glycolysis and the Krebs cycle can be converted to water and drive the production of ATP. Now in the absence of oxygen, Krebs cycle and the electron transport chain, it can't take place, and pyruvate builds up in the cell. It's interesting to note that it can't continue because soon all the FAD and NAD will be reduced. So no FAD or NAD will be available to take up the hydrogen produced during the Krebs cycle, and so the enzymes will stop working. So it's clearly what happens is that in the anaerobic part of respiration, the process of glycolysis is the only source of ATP. So just to recap a bit about anaerobic respiration, very quickly, as you can see, this is very familiar. In fact, it's so familiar it is glycolysis. So 6-carbon glucose being split into 2 pyruvates into 2 3-carbon pyruvates. Now for glycolysis to continue, okay, in terms of the pyruvate being produced, okay, hydrogen must be constantly removed. So in particular the hydrogen must be released from the reduced NAD, okay, in order to regenerate NAD. Without this, then it will be entirely converted to reduced NAD, leaving no NAD to take up the hydrogen newly produced from glycolysis. So therefore glycolysis would grind to a halt. So the replenishment of NAD, okay, is achieved by the pyruvate molecule from glycolysis accepting the hydrogen from reduced NAD. So the oxidized NAD produced can then be used in further glycolysis. So when the pyruvate it's converted to lactate, okay, or lactic acid, you get NAD being produced, and that NAD then cycles back into glycolysis. So let's look a bit more at what happens in plants and animals. So in animals, it's the production of lactate or lactic acid. So they convert pyruvate. This reaction uses reduced NAD by oxidizing NAD. So the NAD is available again to accept electrons and protons. So glycolysis continues because obviously it's not going to gain access to the mitochondria. So if the NAD is not regenerated, even glycolysis would have to stop because there would be no oxidized NAD available to accept these electrons and protons. So the word equation is the following. So pyruvate plus reduced NAD gives lactate plus NAD. So this diagram just quickly shows what happens. So you've got obviously got the glycolysis going on, pyruvate converted into lactate, and the regeneration of NAD to allow glycolysis to continue. So it does cycle around and you're producing lactate. And of course, a small amount of energy in terms of ATP. And just a quick thing about lactate. Obviously, it occurs most commonly in muscles as a result of strenuous exercise, so where oxygen is being used rapidly, and therefore an oxygen debt can occur. Now, your muscles have some kind of tolerance to lactate, but it's acidic and it causes pH changes which will affect enzymes. So it's really important that it's removed by the blood and it's taken to the liver to be converted to glycogen. There are considerable advantages for animals to have this ability because it overcomes a temporary shortage of oxygen. So, and it increases survival, for example, in a baby mammal in the period immediately after birth, and in an animal living in water where sometimes the oxygen levels may be very low. So in plants and some microorganisms and certain bacteria and fungi such as yeast, pyruvate loses a molecule of carbon dioxide and it accepts hydrogen from reduced NAD to produce ethanol. So as you can see, pyruvate plus reduced NAD gives ethanol plus carbon dioxide and NAD. This ability of yeast in particular to anaerobically respire has been used by humans for thousands of years in beer making, in the brewery industry, and also for things like bread production and fermentation, such as grapes, wine production, or other things required in ethanol, used for ethanol. So you can see glycolysis occurring there, okay, but this time carbon dioxide is produced and acetaldehyde is produced, and you get regeneration of NAD again and ethanol being produced. So just to quickly summarize, so in aerobic respiration, pyruvate is converted to either ethanol or lactate, so therefore it's not available for the Krebs cycle. And neither can the electron transfer chain take place. So the only ATP that can be produced by anaerobic respiration is therefore that formed by glycolysis. Let's go through an exam question. So this is an 8-mark question. It's applied in certain sectors and also others it's just recall. So if you look, it says a student investigated the rate of anaerobic respiration in the yeast. She put 5 grams of yeast into the glucose solution right there and placed this mixture in the apparatus shown in the figure. She then recorded the total volume of gas using the gas syringe, collected it every 10 minutes for 1 hour. Question is, explain why a layer of oil is required in this investigation. So the key thing here is about the fact it's anaerobic respiration, and if you look, it's obviously in this flask you're going to have oxygen, so to stop the oxygen getting into the solution there's a layer of oil, so that will prevent oxygen being taken up by the yeast. Now what's interesting is what they'll accept, basically it's the idea of no contact with oxygen, but you need to be careful. You don't just kind of repeat what is in the stem of the question. There's no need to mention for anaerobic respiration because that is mentioned in the stem. Or prevents entry of air. Well, what do you mean by air? You've got to qualify your answer and say, well, it's going to be oxygen. So next question, they give you some results and they show what the student measured So the time, so every 10 minutes they measured it, and the total volume of gas collected. Question says, calculate the rate of gas production in centimetres cubed per gram per minute during the first 40 minutes. So 40 minutes, it was 3.1 centimetres cubed. So what we need to do is first of all calculate the rate. So 3.1 divided by 40, and that will give us 0.0775. Now this is the killer question because it wants it per gram. Now if you remember, if you go back to the stem of the question, it mentions that the student used 5 grams of yeast. So we have to divide that by 5 to find it per gram per minute. So in that case it would be 0.0155. They would accept 0.016 if you had rounded it up. So just be careful with those kinds of questions. Always go back to the stem of the question. Always just check the figures, check everything they've said, so you don't make the mistake of missing that out. Next question. Suggest why the rate of gas production decreased between 50 and 60 minutes. Now, the command word suggest means that there could be several answers here, but if you think about it, yeast is going to need a substrate, it's going to need glucose, but after 50 and 60 minutes it might mean that the glucose decreases. But also you could mention the fact that there's a build-up in ethanol. You could talk about the fact that toxins are built up and the yeast cells have died. So there we have the answers. So you could say that glucose is a limiting factor, or there's an increase in ethanol. You could in fact say glucose is used up. Next question, 2 marks, and it tells you yeast can also respire aerobically. So the student repeated it with a fresh sample of yeast and glucose, but without the oil. All of the conditions remain the same. Explain. So give a reason, give a biological reason why, what happens to the volume of gas in the syringe if the yeast were only respiring aerobically. So it's getting at the fact that aerobic respiration, it would be constant, so it would stay the same level, or be relatively constant, the volume of gas, because the amount of oxygen uptake and carbon dioxide released is the same, relative to one another. So it's the same kind of volume or amount. The final question, it says respiration produces more ATP per molecule of glucose in the presence of oxygen than it does when oxygen is absent. Explain why. Now this question, this type of question comes up time and again in biology, and it's testing your knowledge of the fact of oxygen being the final or terminal electron acceptor in the ETC. So therefore only glycolysis can occur without oxygen, or you could say oxygen is the final terminal acceptor, where ETC provides most of the ATP, or you could, as I said, talk about what happens in anaerobic respiration. Okay, so this brings us to the end of this video on anaerobic respiration. The next respiration video is going to be more about exam technique, and I'm going to particularly talk about the use of a respirometer to measure the rates of metabolism, because that seems to be quite a common style question that comes up regularly. So I hope you found that useful, and I will speak to you soon.